All right. Hi, everyone. Thanks so much for having me. So I wanted to talk a little bit about essentially a paper that just came out. I'll sort of get into details about that. Neuroinflammation in PASC assessed with PET and its correlation with vascular measures. And just as a preface, I sort of agree with what Dr. Hansen just said. And everything that I'm going to describe here, we're also doing in pre-COVID ME/CFS patients. So all of this, is replicated in a larger group because I think it's important to compare. So, when you look at a brain, at least half of the cells you're looking at are not neurons, but rather glia. And that's what this particular study sort of measured directly. This is an image of a microglia, which is the resident macrophage of the brain. When microglia come in contact, either directly with a pathogen antigen or with some marker of inflammation, such as a pro-inflammatory cytokine, they enter a type of an activational state in which they draw in their sort of long, spindly arms and enter this sort of thicker phenotype. Believe it or not, they actually kind of swim around the brain. They're not just sitting there still, but they sort of activate. And when they do, they release a bunch of mediators. So that includes pro-inflammatory cytokines themselves, which are sort of these little triangles here, as well as neuroexcitatory modulators like glutamate, like prostaglandins, bradykinins, ATP, lots of things that excite neurons. In the short term, that's good news because it keeps the neurons near them sort of alive and robust. But in the long term, that can disrupt the sort of normal, delicate balance of the vasculature, cerebrospinal fluid, and neural function. So what we wanted to do is to see whether this glial activation, and it's not just microglia, but there are several forms of glia, whether this long-term glial activation might be part of the long COVID mechanism. So we used, these are sort of the things that can happen here. We used a measure that of what's called the translocator protein, or TSPO, which is upregulated on the outer mitochondrial membrane of glia when they enter this sort of activational state. And it's not a binary, by the way. There's at least 38 activational states that we know of, but this is sort of a somewhat binary. This translocator protein occurs in these microglia, and you can use it in neuroimaging. So you can have a specific type of PET scan that looks for this translocator protein. So in this study, this was sort of a preliminary study, I suppose, that compared a dozen long COVID patients to 43 pre-pandemic controls. We're also replicating in COVID recovered controls, people that report full recovery, but these are people, the controls here were pre-COVID. All of our long COVID patients were August 2021 or earlier, so pre-Omicron strains. All but 2 were pre-vaccine as well, for what it's worth. And all but 2 were relatively mild or moderate cases with only 2 hospitalized. What we did was a little bit, again, what Dr. Hansen just suggested, and I came to the same conclusion as she kind of independently, that we describe these as long COVID patients that fulfill ME/CFS criteria. So in other words, we phenotyped so that we were studying a particular group of patients with diverse symptoms. So our cases and controls were pretty well matched. They were, there was a higher prevalence of women in our case group, as you might expect from a long COVID cohort. And we controlled for sex and genotype, as you're supposed to do in these sorts of studies. We use sort of a standard PET neuroimaging. It's sort of the gold standard for in vivo measurements of neuroinflammation, and this sort of PET technique, sort of pretty standard. We didn't use arterial line here, which we're doing in as many patients as we can now. And then while they're in the scanner, we did a whole bunch more that I won't get to today. Sort of basically standard analysis. So this is sort of the punchline, I suppose, where this is the cases versus controls increased PET signal, meaning increased translocator protein, which we infer to be increased glial activation, or it could also represent glial clustering, where they sort of move to a particular area. So this is sort of the various regions, a little bit sort of on the, for example, on the base of the corpus callosum, the roof of the ventricles, we saw increased signal. Also, where Dr. McAlpine was talking about precuneus, where it says PMCC, that's sort of right in front of precuneus. So it's, this, and again, we came to the sort of same conclusion that it was probably a vascular thing. So what we did was to extract data. I know this is a little busy, but we extracted data from the whole brain, which is all the way to the left, or from those individual brain regions, just to see, is this being driven by a particular brain region? And it seemed like no, there was a pretty good spread where basically all across the cases versus the controls, we saw an increase. It may be a little bit small to see here, but we put the hospitalized cases in open circles just to see. And essentially it was not the case that those were the most severe. There was sort of one that was right about at the median, and the other was a little bit high. So it didn't seem like this was driven by hospitalization acutely. Just as an example, that posterior mid-cingulate that I just measured, we extracted data from that to just take a look. And we were trying to think of, what is the pattern here? This condition isn't really like PTSD, for example, where, the amygdala activates. That's a fear-responsive region. It's a circuitry type of a disorder. We sort of didn't see it that way. So, what might be causing this pattern? Well, as Dr. McAlpine mentioned before, this is an area that we almost don't really have good answers for what it does because it rarely gets knocked out by strokes. It's really well vascularized. So we thought that actually may be part of the story. So what we did was to take blood from our patients right before they got into the scanner and sort of take a look. We looked at 3 sort of different measures: vascular, measures of vascular damage, pro-inflammatory cytokines, and then measures of angiogenesis as well. And just for sort of a little background here, I want you to focus on 3, the subfornical organ. This is a circumventricular organ, so a region of the brain that has very limited blood-brain barrier. We sort of saw at the base of the corpus callosum where there's ependymal cells, again, kind of a window in the blood-brain barrier. It's not as robust as it is elsewhere. We thought maybe that was part of the story of the pattern. And if you take a look, we got signal kind of right in that region. So we thought that perhaps there was a vascular story. And when we did those measurements of the vascular measures of vascular damage and plotted them against the PET signal, we found actually pretty robust correlations. So 0.7, 0.8 correlations are not super common in biology, but we did see that. And again, the hospitalized cases are in open circles, and you see that they're not really the most severe. So for example, fibrinogen correlated, SL-selectin, which was mentioned by both of you guys, Dr. Fraser and Dr. McAlpine, as an adhesion molecule. The whole bunch, basically, there was 6 of them total. So, future work hopefully will start to get a little bit further into the fire and not just the smoke, right? I think a lot of this stuff that we're seeing is sort of the end point. It's not necessarily that this is a disorder of neuroinflammation. I would never argue that. Lots of conditions have neuroinflammation, and the question is, what is driving that? What might be driving that? And this is an example that people have mentioned here earlier of these so-called microclots. This was taken from a patient who visited one of our studies, a 40-year-old woman with long COVID, and these are about 20 to 500 micrometers in diameter. There's somewhat variance in their size and shape. And part of what we're trying to do, again, funded by Polybio, is to crack these open and find out what's inside. It may be the case that they're sequestering spike protein, which has been seen in a couple different studies with the Samoa assay. It's not always consistent numbers, but seems to be a part of the story. So that's part of what we're trying to do. And then we also heard about neutrophil extracellular traps from Dr. Fraser again, and I'm so glad you gave the intro to that so I don't have to. But basically, neutrophils kind of burst open and use the fact that DNA is a long, stringy molecule. They use that molecule almost like a Spider-Man web to trap antigens. And we see that they are occurring a lot in long COVID blood. We're also doing this in pre-COVID ME/CFS, which I'll talk about in a moment. So this is an example of a stain in blue for the extracellular DNA that represents the extracellular trap. And we looked for evidence of platelet activation using different fluoroscopy colors, and found that they co-localized quite a bit. So again, that's smoke. What's the fire? We're not quite sure, but they're sort of overlapping. The activated platelets and the activated neutrophils seem to be co-localizing, so maybe they're responding to the same thing. These are some, part of what we do, which I think is pretty interesting, largely led by Daniel Laremia's lab, is neutrophils are sensitive. They're very sensitive markers. They're sort of thought of as the pawns of the immune system, but their function is really disrupted if you freeze them and thaw them again. So what we do is to take them when they're still alive, fresh, within 3 hours of taking it from the participant and looking at the function. So what we did here was to look at the neutrophils. In this case, it's actually plasma from a patient put onto neutrophils of a healthy person. And you can see as they burst their neutrophil extracellular traps, sort of release their traps, the plasma of the patients cause this bursting in healthy neutrophils. So we're quantifying that and sort of measuring. Again, one of the things that we measured as correlating with neuroinflammation was that SL-selectin, which is something that would cause a neutrophil to bind to the vasculature and get pulled into a tissue. So that may be the source of the neuroinflammation that we're seeing at a cellular level. This is another example where, hopefully that will, is my video coming up? Oh, really? I'm only 11 minutes. These are cool videos. Hopefully we'll get it to work. So there's another video of live neutrophils where essentially we just put them into a maze and let them swim. A neutrophil of a healthy person shouldn't swim very much. Can you get it? You can just show the sick person if it works. Okay. You got to trust me that these are cool videos. So I'll just describe it in words first. So we start with a healthy control and show the neutrophils swimming through this maze, and they just swim spontaneously. And in a healthy person, they barely swim at all. They sort of don't scoot around. Probably not coincidentally, that wouldn't be particularly energetically demanding. But then when we take, in this case, a different 40-year-old woman and put their neutrophils into that maze, they swim quite a bit. They're sort of ongoing, not just one or two neutrophils scooting, but a whole bunch of them swimming around at once. So again, that's probably smoke, not fire. So, okay, great, thank you so much. So this is a healthy person, and you see a little bit of scooting, kind of not that much, and you'll see why it's not that much in a moment. Can you do the next one, or can I try to do the next one? Okay, so now this is a patient. So quite a bit more, as you can see, right? So they're sort of activating. That's not mine, by the way. I don't know why that, it's not mine at all. I had nothing to do with that. I don't know. Someone's setting me up. It's totally not mine. So what you're seeing here is spontaneous migration, which is to the left. Again, we compared to pre-COVID ME/CFS, and this is to Dr. Hansen's point. We have examples where it's similar, and we have examples where it's different. And for that matter, not all patients show this, just most do, or many do. And then we also showed the spontaneous netosis, in which the ME/CFS and the COVID were not different, and then the PMA simulated where the ME/CFS was not as robust as the long COVID. So these are sort of areas to compare. And then just another sort of future work that we're working on just now. I'm just excited about it because we were working on it yesterday. We're going to try and measure the bone marrow of the skull to see if perhaps there's inflammatory activation there. We know that that's a source of immune cells. There's good bone marrow work happening right now that, we may start to see that bone marrow is part of the story in this condition. So I'm right about it exactly at 15 minutes, so thank you very much.